Good evening. Today I'm going to be presenting a research done by Jacker et al. And it's trying to tackle the question of, is Alzheimer's disease a prion disease? Prion disease is caused by prion proteins that gather around a neuron, and it is thought that it allows too much fluid to go into the neuron, eventually killing the neuron. The disease spreads throughout the brain and forms these dark veins, as we can see in this picture. Prion disease is caused by a mutated form of prion, of a prion molecule, and the prion molecule is a protein that is generated by the PRNP gene and is vital for the survival of the organism. They're more frequently found in the nervous system. When a normal prion mutates into the mutated prion form, what happens is the mutated form can no longer be used by the organism. So the mutation renders it unusable biologically. And this molecule then travels around the brain, and it has the ability to change or mutate every prion molecule that it encounters that it's of its same kind. So in essence, it goes throughout the brain and just starts mutating all the existing prion molecules that are in the brain, spreading the pathology. The way this protein mutates is mostly by a random mutation. So it is believed that about 85% of cases of prion disease are caused by random mutations. There are cases in which a gene can actually be passed on from generation to generation. There is an example of that in the human population. There's a disease that's called the fatal familial insomnia, and there are about 40 families known around the world that have the disease. So it's very rare. Another way that prion diseases can be transmitted is through the environment. So there have been cases in which the disease has jumped from species to species, and also from an individual to another individual within the same population. So how disease can jump from one species to another? It's because there are highly conserved genes, and this gene, the PRNP gene that codes for the prion protein, is considered a highly conserved gene. So it is thought that the close relative of this gene, of this protein, doppel, was likely present in the last common ancestor of present-day tetrapods. And so as you can see, tetrapods gave rise to many, many different animals. Among them, you see mammals, also reptiles and birds, and amphibians. And so if you have a gene back when they were tetrapods, and this gene has been passed on and has not disappeared, it's just passed on to Amniota, to Mammalia, to Lepidosauria, to Aves. So this is a gene that is producing a particular protein. So in this case, if you have a mutation of on that particular protein, such in the case of prion diseases, and this protein exists in another species, and it has a function in another species as well because it has been preserved, then what happens is that if an animal experiences a mutation, and another animal is exposed to that mutation, to that mutated protein, it can also affect the other species because that other species has the functional protein in their organism that this mutated protein can actually start mutating, in essence, spreading the disease. And it's very similar to what has happened with the coronavirus right now, where a virus has jumped from a bat to a human. So bats are mammals, and obviously it looks like this virus is acting on a particular protein that is shared between those two species. And so that is how coronavirus has been able to jump. Prion diseases have been known to exist for hundreds of years. The oldest prion disease to be known to exist is scrapie, which affects sheep and goat. Um, it is believed that a spontaneous mutation happened at one point and then it's just being passed on from one individual to another. Um, the one of the clear signs that an animal has the disease is that they start scraping themselves or scratching themselves around against fences, posts, stones, anything very compulsively, as if they have an itch that they cannot really get rid of. Eventually, they get basically they get rid of all their wool this way. They damage their skin, and this disease progresses into paralysis and eventually death from starvation because which seems to be a common sort of theme between degenerative diseases is the loss of muscle function, and because of that, you cannot swallow food or chew it really. Another famous case of prion disease was mad cow disease, which basically experienced an outbreak in England in the 1980s. This disease was actually transmitted to humans in England, and there's documented cases of more than 200 people that acquired the disease after eating meat. One of the characteristics of prion proteins is that even when you cook them, they have been known to be transmitted. So heat does not necessarily seem to render them inactive. If you donate blood in the United States, you are asked, have you lived in Europe between 1980 and 1996? Or have you been stationed there? Many people were stationed there at the time. And if your answer is yes, you cannot donate blood here just to protect the blood supply. In Europe, they have a test, so they actually administer a test. Another well-documented case of prion disease, this time in human population, um, uh, took place with the Kuru people of New Guinea. In the 1950s, there were around 35,000 individuals, and they were experiencing around 200 people dying of the prion disease every year. And the reason why they had this higher frequency of prion disease is because they have a ritual in which they're in the funeral, they kind of eat the a person that is deceased because they believe that it was better that the people that loved the person ate them as opposed to the ants or other animals. And so the way they did it is the men consumed the flesh and the women consumed the brain. And so the disease was passed on more to women than to men because the disease is more concentrated in the brain. And here is the Kuru disease as seen in the cerebellum. You can see these veins, they're like gold veins in a gold mine. If you've ever seen a gold mine, it's just like this. It's just little veins of gold in the middle of all stone. And in here, you can see that dark pathology going through the brain. Here you can see the damage that the prion disease is causing the nervous system. It causes a spongy-like sort of consistency. And so I guess if you had to have a zombie disease, I guess the prion disease could be it. So let's dive into the study. So this study basically was tackling the question, could Alzheimer's disease be transmittable like a prion disease is? And so what they did is they transplanted tissue from a deceased human as well as a deceased mice brain to a host mice hippocampus. And they saw if there were any amyloid beta aggregates developing. So basically any sort of sign of the pathology of Alzheimer's disease. They used the standard model organism for Alzheimer's disease, the APPP23 and APPP-S1 mice. I'm going to abbreviate this to P23 and PS1 mice. The Alzheimer's disease, just briefly, develops when an enzyme cuts a particular protein at the wrong length and it makes it too long. So it makes it 40 or 42 amino acids in length. This particular cut then cannot be used by an organism and accumulates in plaques like we see here in the brain. Here's an example of tissue from Alzheimer's. showing the amyloid plaques right here in pink that the study looks at. So the study basically took extract from, we said, deceased patients, and it transplanted it into 5-month-old P23 mice that had not yet developed the pathology. P23 mice develop pathology typically at 9 to 10 months old. So here in picture number A, you had extracts from a deceased human Alzheimer's disease patient during their autopsy. They took some tissue and they created 10% extract, which was then implanted into the mouse. And 4 months later, you can see that the pathology developed. Same thing happened with the next experiment. You can see in picture B that the extract from an APP23 mouse was implanted into a younger APP23 mouse, and the pathology actually did develop. Next, they took an extract from a 95-year-old control patient that did not have Alzheimer's disease, and you can see that the pathology did not develop. And it was interesting that it developed a little bit of pathology which mimicked exactly the same pathology found in the 95-year-old patient. Here in D, you can see they took extract from a wild-type mouse and they placed it into the P23 mouse, and no pathology was developed. E was just a control substance. They put in the PBS injection, which is just a saline sort of salty, watery injection that you can put in the brain. It doesn't have any effect, but it's just to see if the tissue reacts at all in the host mouse, and you can see that nothing really happened. And then lastly, they took the APP23 extract and they placed it into a wild type mice. And as you can see in this case, the pathology did not develop, where it did develop when they put it into the P23 mouse, so model for Alzheimer's disease. But in this case, wild type mouse did not develop the disease, which shows that perhaps there is something in the host brain that is necessary in order to develop the pathology. So in order to do that, they just asked the question: does amyloid deposit this amyloid deposits, are they dependent on the host, or are they dependent on the agent? And so here they use the P23 and PS1 mice to test this hypothesis, that both the agent and the host play a role. So here you can see that, for example, in picture A, the PS1 extract was placed into the P23 host, and it created a pattern very similar to what a P23 host develops naturally. If you can see in picture C, a P23 extract was placed into P23 host and developed the pathology that naturally P23 would have developed. And if you can see picture A, the host sort of pattern was also developed in here, but it was a little more diffused as per the PS1 extract. Same thing happens with picture G, where you can see the P23 extract placed into the PS1 host caused sort of, you can see almost the P23 shape, but it's very, very diffused. It's almost like the host PS1 mouse is actually diffusing sort of that deposition, just like you can see in picture E, where PS1 extract was placed in PS1 host. You can see that the positions are much more diffused. And so the same you can find in G. You can still find the shape of the pattern from the P23 pathology, but is much more diffuse, such as in the case of the PS1 pathology in picture E. So we can conclude this from that the agent and the host biological properties do contribute to the spread of the disease, and not just one or the other. The next thing they asked is AB needed for aggregates basically to develop? And so what they did is they took an extract and they depleted it using immunodepletion technique. and they injected it into a host, and no pathology basically developed without the AB. Then they took the extract and they left the AB, they left it alone, but they added beta 1, which is an AB blocker. And basically, the experienced pathology, but as you can see, much less in picture E. You can see versus picture D, which would have been without the beta 1. With the beta 1 in picture E, much more reduced. Then they took the extract and they um, added passive immunization. So this is where they take basically antibodies from a host that has experienced the disease and they transplant it to another host, just like they did with Trump coronavirus. They took the antibodies and they treat him with the plasma of those antibodies from a different patient. And in this case, the pathology did not develop in picture H. Then they took the extract and they added formic acid, which actually rendered the extract basically ineffective. As we can see in picture K, the pathology did not develop. And then they exposed the extract to 95 degrees Celsius boiling water, and then they injected it into the brain, and basically the pathology was visible, but it did not develop as much as it would have if it had not been boiled. So the next thing they did is a particular feature of prion diseases is that if you use a synthetic form of the prion protein, it does not, and you inject it into a host, it does not develop the disease. So synthetic prion proteins do not develop disease. So they wanted to see if it's the same case with Alzheimer's disease. So they used synthetic AB extracts and they implanted them into host. And as you can see from pictures C and D, none of the extracts, neither the 140, the 142, or a combination, were actually effective at developing the disease as compared to a control in picture B, which was the regular extract without the synthetics. So synthetics basically did not work in this case. So here they just found that the exogenous induction of AB deposition in vivo is time and concentration dependent. So that if you use a 10% concentration, you get more disease basically than if you use a 5% concentration. And the pathology appeared in this case at 2 months and grew exponentially after that. So the conclusions for the study basically was that Alzheimer's disease does in fact behave as a prion disease. Alzheimer's disease transmittability depends on both the agent and the host. And Alzheimer's disease is transmittable, but it isn't clear if it's contagious like some prion diseases are. So it may not be contagious through, let's say, saliva or bodily fluids, like for example HIV is. That has not been proven, but it is certainly transmissible if you use direct exposure to nervous system tissue. And so if you come across contaminated tools in an operating table, you could become contaminated with Alzheimer's disease pathology. Also, the synthetic AB failed to induce AB depositions, which is similar to prion disease. And interestingly, they were able actually to use the beta 1 as a blocker for AB and almost like acting as a vaccine. So it would be interesting to see if there could be a vaccine at some point for Alzheimer's disease. There's a lot of research right now taking place around this idea of diseases that can behave like prion-like diseases. And a recent study by Junker Silva in Brazil has found that there's actually certain types of cancer that do behave like prion diseases. And in this case, for example, there's a cancer caused by a protein called P53. This protein basically acts as a tumor suppressor normally, but when it mutates, it actually changes function and it basically lost the original function, it gains a new function, which is actually promoting tumors. So, and so there's some research being done on finding that some types of cancer can actually be also behaving like prion diseases. And so this concludes my presentation. I hope you enjoyed it and have a good night. This is my bibliography. Ciao.